Welcome to Lecture 11. The topic is What is a Pathogen and Understanding Infection? This is the first of a two-part lecture. In this lecture, we're going to talk about biological associations or relationships in the context of infectious disease, talk about parasitism as a lifestyle, and microbes as disease-causing agents. We'll be reviewing Koch's postulates as we do that in preparation for a lab on the topic of Koch's postulates, which will begin next week. We will also talk about microbial mechanisms of disease. We'll talk about the concepts of pathogenicity and virulence, and the defensive and offensive strategies that pathogens utilize. We'll also talk just a little bit about virulence in a few non-bacterial pathogens as we go along. And then we will look at the terminology used to describe the stages of a microbial disease, or maybe more accurately, the stages of an infectious disease. So when considering the relationship between a microbe and a human host, since we're focusing on humans, it's important to remember that it is a relationship. So let's define a couple of terms. Let's define the term symbiosis as living together, or the association between two or more species. And symbiosis can be considered as an umbrella term into which fall the terms mutualism, commensalism, and parasitism. Mutualism means that you have a relationship between two or more species that benefits all of the species involved. Commensalism means that you have a relationship between two or more species, but it's unclear whether there's any harm or whether there's any benefit. It seems like they need each other, but the relationship is not well understood. In the case of parasitism, you have a relationship where one is clearly benefiting and the other is clearly being harmed. In the case of infectious disease, that is an example of a parasitic relationship. In that case, the biological association between host and microbe is definitely tipped in favor of the microbe and not in favor of the host. But these relationships are very dynamic, and so they can change, as the conditions for the host and the microbe change over time or due to a variety of circumstances. Let's consider an example of how a relationship between human host, and microbe can change. Let's think of the example of E. coli, which is normally found in our large intestines. So we all have E. coli in our large intestines, and it's a very useful part of our microbiome. We provide it with food, mostly in the form of sugars, and in return, it provides us with vitamin K and several other benefits, including the ability to digest food properly and to resist many pathogens. So that's an example normally of a mutualistic relationship where the host and the microbe would be existing in, we won't use the term balance because it's not really accurate, but both are benefiting. It's mutualism. Now consider the example of let's think of a time when that relationship might change into a parasitic relationship. And an example of that might be if E. coli makes its way to the entrance of the urethra. And that can happen as a result of contact during sexual intercourse, or in children if they're being cleaned inappropriately during diapering in very young children. So let's say that the E. coli makes its way to the entrance of the urethra, or even a little bit into the urethra, which is essentially the tube in the body out of which urine normally comes. If that E. coli establishes itself in the urethra and starts to grow, then that is the source of a urinary tract infection. And an infection means that the host is being harmed. And so in that case, the E. coli and the human host would no longer be living in a mutualistic relationship, but instead would be living in one that is definitely parasitic, where the E. coli is favored, but the host is not. There's no benefit to the host to that infection. So that is the result simply of E. coli being at a location in the body where it's not supposed to be, that being the urethra. There are many other circumstances that can result in changing the relationship from either mutualism or commensalism over into one of parasitism. Another example might be immunosuppression. For example, someone that was undergoing chemotherapy or was immunosuppressed because they were they were starving, or they had too low nutrients, or their system was run down for some other reason. In that case, the normal commensals or mutualistic organisms might start to multiply unchecked, leading to an infection. So there are many examples of when this can happen, but it's important to remember that we live in relationship with the microbes on and within us, and that many of the organisms that can act as pathogens are actually part of the normal human microbiome, and it's simply that they're acting as pathogens because the relationship has changed. Now, there are, however, some examples of organisms that are always pathogenic. For example, Bacillus anthracis, which causes anthrax disease, or Yersinia pestis, which causes what we call the plague. We never live in symbiosis with those organisms, or in mutualistic relationships with those organisms. We always live with them in situations that are considered parasitic, where they're acting as pathogens. Lewis Thomas, a famous medical doctor and microbiologist, a specialist in infectious disease, described what I'm talking about with the quote shown here. Disease usually results from inconclusive negotiations for symbiosis, an overstepping of the line by one side or the other, a biological misinterpretation of borders. This figure shows the location of where many microbes are often found in the body, although that's not entirely true. So the urethra and the conjunctiva, which is the lining of the eyes, would normally have very, very few microbes. They're very clean environments from a microbial perspective. But you could have infectious disease. If we go back to the example that I just discussed on the previous slide, E. coli, which is normally found in the large intestine, if E. coli moves from the large intestine to the urethra, and that happens more frequently in women than in men as a result of the length of the urethra. Women have a much shorter urethra, so they're much more susceptible to infection in the urethra, but that movement of microbes can be one of these overstepping of the lines by the microbial side, leading to infection and a parasitic relationship, that being infectious disease. So the focus of today's lecture is on understanding, what makes a pathogen a pathogen. And before we really embark on that, we should go back and review the idea of the germ theory of disease, which I introduced earlier in class. So this is the idea that was first recognized in the 1800s, which was the first golden era of microbiology. During the 1800s, sanitation or hygiene was recognized as playing a key role in disease. So people were really figuring out that clean water, clean food, and clean living situations were ways to reduce the possibility of infectious disease. The idea of quarantine was also quite common during the 1800s. And in the 1800s, that's when Louis Pasteur and Robert Koch devised the germ theory of the disease, which is the idea that microbes cause infectious disease. And Koch came up with his postulates to prove that a specific microbe could cause a specific disease. Let's review Koch's postulates, which are shown in figure and text form here. So the four pieces of logic here, and they go in order, this is very linear logic, are association, isolation, causation, and re-isolation. And remember that all of this depended on the ability to grow microbes in pure culture in the laboratory. And that was first devised using agar, something discovered by Fanny Hess. It's a solidifying agent that allows us to have plate cultures and plate cultures of bacteria. When you grow bacteria on plate cultures, you can separate one from the other. So Koch's postulates begin with association, which is the idea that all diseased individuals will have the microbe of interest. Then they move on to isolation, which is the idea that you can grow that microbe in the lab in pure culture, so isolated from other microbes. And then the idea of causation, which means that if you take that microbe and you inoculate it into a healthy host, here it's a mouse, okay, then you will see that new host develop the same disease, and you'll know it's the same disease because it has the same symptoms. and that you can then re-isolate the microbe from the newly diseased individual, grow it in the lab again. So here we grew it in the lab in the beginning, and we're growing it in the lab again, okay? And you can then confirm that it is the same microbe. So you need to confirm that these are the same microbe in order to close this logic loop. Those are Koch's postulates. Using those postulates, Koch and Pasteur proved that Bacillus anthracis causes anthrax disease. They proved that Vibrio cholerae causes cholera, and they proved that Mycobacterium tuberculosis causes TB, as well as many other pathogens still important today. These were the years when Koch and Pasteur and other scientists used this logic to determine what pathogens caused what diseases. Now the reality is that it's not always simple to grow a microbe in the lab. And the most obvious example of that is growing viruses. Viruses cannot grow without cells. They are intracellular pathogens. And so growing them in the lab can be very, very difficult. And so that makes using Koch's postulates exactly as I just described them more challenging. So we have some adaptations to Koch's postulates that are used today, commonly in labs and by researchers studying infectious disease. And since we have gotten good at working with isolating DNA and sequencing DNA, we have several molecular adaptations of Koch's postulates. So a similar way of going about Koch's logic is shown in this slide. First, you identify a gene or a gene product that is responsible for virulence. Virulence is the ability of a pathogen to be pathogenic, to be harmful to a host. It's a measure of how harmful a pathogen can be to a host. And virulence is ascribed to a set of factors which I'm going to discuss later in this lecture, most of which are protein, and therefore they're encoded by genes. So you identify a particular gene that is responsible for virulence, then you show that that gene is present in a microbe, in a pathogenic strain of a microbe, show that it is not present in strains of the same microbe that are not pathogenic. And here you might want to think about the example of E. coli. E. coli comes in hundreds of different strains, most of which are not particularly pathogenic, but some of which are very dangerously pathogenic. Those that are dangerously pathogenic have some specific virulence factors that make them good at infecting and harming hosts. And there are genes for all those virulence factors, and those genes are found in the deadly E. coli, and they're not found in the non-deadly or non-pathogenic E. coli. You carry on with this kind of work by showing that if you disrupt or mutate the gene, you reduce virulence. And then to get a little bit more proof, you show that introduction of a clone gene into a non-pathogenic strain can turn it into a pathogenic strain. And you also have to show that the gene is expressed, in other words, it is utilized in living cells to make a protein product. And finally, you have to show that a specific immune response in the host occurs in response to the product of the gene and that this protects against disease. So you have to show something about the relationship between the host and the gene product of interest. All right, so let's move on into talking about microbial mechanisms of disease. So the figure on the top shows the varying levels of infectious disease that one can have, ranging from being almost normal, perhaps just a little bit tired or under the weather, that's the subclinical level, all the way through to extreme infectious disease that causes death or hospitalization. Let's define infection as the multiplication of microbes on or in the body without producing definitive symptoms. So this is typically the subclinical stage. You might have some antibodies present as a result of your immune system recognizing that a pathogen is there, but you're not feeling terrible. You're mostly normal at this particular point in time. That's the beginning of infection. And as the microbes multiply on you, you will reach whatever level of disease is typical for that pathogen and yourself, whether you're healthy or not healthy, the degree to which your immune system is able to fight it off. Let's define disease as the impairment of health to some degree. And so let's ask the question now, what dictates the progression of infection into the result of actually impairing health, into actual disease? And this can be actually thought of in very mathematical terms. If we say that D is disease, which is the impairment of health to some degree, and dose is N, lowercase n, that is the number of microbes that a person is exposed to or that a person has on them within the moment that you are considering. Let's use the term V for virulence, a measure of how dangerous a microbe is. And let's use the term R for resistance, which is a measure of how well a host can fight back. Then you can use that equation shown there, that is D equals N times V, all divided by R. So your level of disease is a function of the number of microbes that you have times how virulent they are, divided by resistance. So let's think a little bit here now. If you had been vaccinated and therefore you had an immune response that was highly effective at clearing a pathogen, then even if the N and V terms are really high, in other words, you were exposed to a lot of a pathogen and it was a really dangerous pathogen, your resistance would be very high. And since resistance is the denominator, then you would expect your D value to be very, very low. In contrast, if you hadn't been vaccinated, and so your resistance was very low, and you were exposed to a certain number of microbes with a certain virulence, then your R term will be very small, and so your disease, your overall level of disease will be much higher. Let's consider the idea of infectious dose for a moment now. So infectious dose can be given the term N. This relates back to the equation that we just described. The infectious dose is the number of organisms required to make an individual sick, to infect an individual. And since individuals are quite variable, and it might take 10 of a microbe to make me sick and 20 of a microbe to make you sick, then this actually has to be worked out somewhat mathematically using experiments in the lab. Most typically, what scientists calculate is something called the ID50 value, or infectious dose 50. And that is the number of pathogenic microbes at which 50% of hosts will be infected. You obviously can't do these experiments in humans, but you might do them using human cells in culture, and you might look to see what dose it took to infect 50% of the cells exposed, or you might use an animal model such as mice, or you might determine the infectious dose or the ID50 number by looking at or using mathematical modeling and looking at a scenario that already happened in humans, trying to figure out about how many microbes they would have been exposed to and counting to see how many people were exposed to how many microbes and how many of them got sick. So that's the idea of infectious dose, and it's something that really is pathogen specific. So for example, Mycobacterium tuberculosis, which causes TB, is highly virulent. So if the ID50 value for Mycobacterium tuberculosis is 10 bacterium, which could easily be found in a little droplet from somebody sneezing on you. Vibrio cholerae, in contrast, which is the organism or bacterium that causes cholera, that's a much less virulent strain because it takes on average about 1,000 bacterium to infect an individual, so a much higher dose, much higher N number. So this is something that really varies by microbe, and there are no sweeping patterns to it. It's just simply a quality of a given pathogen that you have to understand in order to know about that particular pathogen. So with that, I will now ask you to move on to part 2 of this lecture.